Hello guys, welcome to my YouTube channel. My name is Manas and today we're going to see construction of cDNA libraries. Why is this method important? As we know, in Human Genome Project, which was started in the late 1980s by Watson from the famous Watson and Crick duo that won the Nobel Prize for construction of DNA molecule. So what did Watson think is a human feat, a human miracle of construction each and every nucleotide in human genome, entire human genome. He wanted to sequence this thing to know which particular genes are causing the diseases, which particular genes are helpful to us, which are not helpful to us. I mean, all of the genes are obviously not encoding for a specific protein that might be helpful for the human body. So for this method to achieve, we obviously need this cDNA libraries. Now, how do we isolate cDNA libraries? Is the first step in it is isolation of mRNA. mRNA, as we know, is the messenger RNA that encodes for a protein, which is the method called translation. We do not actually isolate mRNA. We isolate mRNA from RNA because this molecule, it is very prone to degradation. It degrades very easily due to some minute changes in pH or minute changes in the temperature. Next, we shall purify the mRNA. Now, even though we do isolate this mRNA from RNA, there might be some components that are not useful to us, that acts as what do you call it, garbage, which do not yield a pure mRNA molecule. And for that, we use affinity chromatography. By affinity chromatography, we can elute out pure 99.9% mRNA. Next step is cutting the mRNA from its 5' cap and 3' tail. As you know, an mRNA, suppose this is an mRNA, at the end of its, at the 3' end, at the 5' end, at the end of the 3' end, there are poly-A chains. There are poly-A chains, and at the end, in the beginning of the 5', there are particularly guanidine caps. For isolating pure mRNA that can yield our cDNA, we have to cut these guanidine and poly-A tails. We cut these poly-A tails by exo3. As we know, exo3 is an exonuclease, and it cuts from the 3' end of any DNA. And to cut the guanidine caps, we use acid pyrophosphate. Acid pyrophosphate enzyme. We isolate this acid pyrophosphate from BAC and TAC. Tobacco acid pyrophosphate or bacterial acid pyrophosphate. The next step is converting this mRNA into single strand DNA. This is single strand cDNA, which is also called single strand cDNA. Now, you may say, how do we convert an mRNA to DNA, which is not possible in cells because usually we see DNA converts to mRNA by transcription method. But how do we convert mRNA to single strand DNA? We use, yes, correct, RT polymerase. Here RT stands for reverse transcriptase. Reverse transcriptase. It converts an RNA to DNA. But mind it, this RNA will convert to single strand DNA. We obviously need a double strand DNA to consider it as a DNA which can be used to make a library of a particular organism. Let us see how do we do that. Yeah, so we were at the step of getting single strand DNA. Now we got the single strand cDNA. But mind it, imagine this is the DNA. The mRNA is still currently attached to it. It has not left it. How do we break this bond? We break this bond by adding RNase H. RNase H. Why particularly RNase H? Because it does not degrade the RNA, it just breaks this. It just breaks this RNA and DNA hybrid molecule. So we get a single strand cDNA, more of a purified molecule. Now, how do we make a single strand cDNA to double strand cDNA? We'll see that. Now, single strand cDNA, we have to make it to double strand cDNA. And this is only possible through PCR, that is polymerase chain reaction. What do we do in PCR is, as we know, we include primers and polymerases. DNA dependent DNA polymerases, because we are using a DNA template to make another complementary strand of DNA. So this DNA will get its another complementary strand which will make a complete double-stranded DNA. We use PCR technique to make a double-stranded DNA. The next step is inserting this double-stranded DNA and taking a gene of vector. Even though we have isolated the entire mRNA into this DNA, we might need a small part of this DNA as our gene of interest. We'll take a small part gene of interest as gene of interest. We'll cut it out by endonucleases and we'll do recombinance into suitable vectors. I mean, if you have to take a small part of the DNA as gene of interest, you may take it, or you may take the entire double-stranded DNA as gene of interest, because there are some vectors which can also take up genes that can have a size of 1 MB, that is 1000 kilobase pairs. Yeah. So we'll perform recombinance into suitable vectors, possibly M13, MP1 or M13, MP3 vectors. After recombinance, we'll have screening of gene of interest. We'll screen the gene of interest based upon the marker site that this vector has, M13, MP1 and M13, MP3. We'll put a nutrient medium and an antibiotic to see which colonies are growing. If the antibiotic is really present in the M13, MP1, it will grow colonies. When it will grow colonies, we may say that this gene of interest is present in the vector. The next step is in this. So the next step after recombinance of the vectors and screening of the gene of interest, if it is really present in the vectors or not, we'll transfer it to a host system. Now, please do keep in mind is that when we select the host system, it must be compatible with the gene of interest that is being taken and the vector that is being taken. If the gene of interest and the vectors are prokaryotes, we must use a host system that is also prokaryote. But due to recent advancement in science, due to recent advancement in biology, we can also take a mix and match. Even if the vector is a prokaryote, we may use a host system that is eukaryote. And that is how a famous example for this one is using E. coli to make humulin. Humulin, as we know, is the alternation of insulin that is being made in E. coli. After transferring it to the host system, will make it amplify, will make the colonies grow. Colonies grow for further experimentation and for further use. The next step is storage of this cDNA. Now, as I said before, if you want to experiment the cDNA in a very short time, we may use slant method. Or if we want to store the cDNA for many years, possibly 5 years to 10 years, we'll store it in liquid nitrogen. And the slant method can store the cDNA for some months. So that is it, guys. These are the possible steps for construction of cDNA libraries. I hope this video will be helpful for your future endeavors. My name is Manas. Peace out.
Adbhut! Pack up, cut.
Yes.